We began working with the SMA Foundation, and foundation basically was started by a family who has a child with SMA, and their mission was to identify a treatment to help their daughter. It was that partnership that allowed us to get going. They were part of the team.
Alternative splicing is a biological mechanism that happens with many genes in the human cell. The way that the genes are organized in humans is that they have interrupted sequences. The final coding sequence of the messenger RNA consists of small, sometimes not so small, fragments that need to be joined. The process of joining these fragments, called exons, together is splicing.
When we initially set up our SMA program, we had 3 different programs that we were testing that we thought could be viable to see if we could affect splicing. Initially, and we have a library of about probably 300,000 molecules, and we screened all of those, and we had hints of activity. We first were able to identify molecules that affected splicing, then we built the selectivity and specificity into that, and then we built the pharmaceutical properties into that. We get all of those properties into one molecule that we then say we're going to move and test into the clinic.
In order to identify and develop this SMA compound, there were multiple teams at PTC that were involved in making it happen. They included biologists, chemists, pharmacologists, toxicologists, formulation specialists, and many other people. They actually all work together with the goal of identifying a compound that is both effective and safe. But of course, all their work could not have been possible without the support from so many other people at PTC.
Most traditional medicinal chemistry programs initiate with a high throughput screen, and the SMA program in splicing was no different. We screened our library of compounds, and we identified a number of very weak hits. From there, the effort was quite extraordinary to figure out if one of them had true splicing activity. I still recall the day when a set of analogs were made, when we recognized something a little different. Those analogs had an activity that we hadn't seen in other hits. And as a team, coming to the conclusion that we had found something special, we put all of our effort toward those molecules, and in a matter of several months, we were able to improve the potency 100-fold. And this led to molecules that for the first time showed SMN protein increase in patient cells.
One of the things I'm most proud of, beyond identifying a drug, is the team. It really demonstrates how a small group of people could have a profound effect on the lives of so many people, patients and their families. And they did something that they're going to be able to tell their grandkids that they did something important in life and changed the lives of a lot of people.
Just thank you to everyone at PTC who's worked so incredibly hard and have put your lives and your energy and your time into this. It means you're saving lives for SMA patients, and I don't think that anyone will be able to express the gratitude everyone feels, but at the end of the day, I think just simple thank you for sticking it out and pushing through and, having hope and tenacity to work on SMA when a lot of people just simply didn't. So a huge thank you.